proteomic imaging using our ToF-SIMS system, which we commercialize and call multiplexed ion beam imaging, or MIBI. And so my 6 years at Ionpath, I've actually had a few different roles. I started out as a data scientist. As Wah mentioned, I spent a lot of time in my PhD and postdoc doing data analysis and algorithms for SIMS images. So I did that for 3 years. Eventually, back in 2020, I took on a new role at Ionpath in which I was the head of our field applications team, so directly supporting our MIBI scope customer base who had purchased our system and had been using our platform. And then earlier this year, I switched to a different role as director of product development, where I now oversee pushing our technology forward and to the next level, including a new services offering that we launched last week, which I'm going to talk about as well. And so a quick little overview of my presentation to you today in this webinar is to give you an introduction to the spatial proteomics with ToF-SIMS using the MIBI platform. That is what we commercialize here at Ionpath. So I'll walk you through why we do what we do, what the use case it meets, and how we've designed our platform to accomplish that. And then I'm going to go through a case study from a MIBI data set to show you how we go from tissue sample to proteomic spatial profiling, show you how we stay in the image, stay in the samples, acquire the data, and then do the data analysis to really figure out the spatial relationships of the cells and the cell types within the tissue images. I'm going to highlight a few MIBI applications from some of our customers at Stanford University, where we were spun out of. And then I'm going to talk to you about Ionpath's new MIBI Plus, which is an all-inclusive offering for studies of any size that's just, like I said, just launched last week. So really, to understand what we're trying to accomplish here at Ionpath is to really understand how the tumor microenvironment is a diverse, encapsulation of a piece of tissue. Sorry, excuse me. Tissue microenvironment encapsulates a particular region, and some call it the most reliable readout of disease state and patient response. When we think about imaging cells, we can see a cell in a particular place and know maybe what its cell type is and what its cell function is, but that really only paints a particular picture of what's going on in that tissue, whether it's healthy or whether it's diseased. And really what we need to do to understand exactly what's going on is to understand the entire microenvironment itself, because as we know, tissue is a heterogeneous environment made up of many different cells and cell types. And often what we find is it's not just what cell types are present within a tissue microenvironment, but it's also what their spatial relationship is to one another or to cell types of different functions. And so really to understand disease better, we need to understand, or we need to appreciate the tissue's function as an ecosystem themselves, and that health and disease are emergent features that depend on the cell composition, the diversity of the cell types, those cell locations, and the function of those cells. And you can imagine that this is pretty complex. And so in order to appreciate the number of different cell types there are within a particular microenvironment, we really need a highly multiplexed technique for that. And when I say highly multiplexed, I mean we need to be able to simultaneously analyze many, many different types of cells. And to do that with proteomics, we need to measure simultaneously many, many different proteins. And so here's just an example of a number of different types of cells that are within that can be found in a particular tissue. And you can see that there's 8 main classes, but there are many different markers for each one of those classes that tell us what type of subclass those cells may be, or whether those cell types are activated, whether they're actively fighting an infection or a disease. And realistically, we need to analyze 30 or 40 of these different proteins at the same time to build this entire comprehensive picture of the tissue microenvironment. And so we need a highly multiplexed imaging platform for this, where we have at least 20 markers for immune cell phenotyping and cell function on a particular sample itself. We also need a high resolution. We want to see where single cells are. And so we have to have subcellular spatial resolution so that we can resolve and we can image individual cells within that tissue microenvironment. And so we need a minimum of 800 nanometers spatial resolution in order to segment the tissue into individual cells. We need a high sensitivity and wide dynamic range because many of these different proteins are expressed at different levels. And so to be able to simultaneously image them, we have to be able to image high concentration or highly expressed proteins at the same time as low expressed proteins. And we also need high reproducibility as, well, any other analytical technique does. And so the breadth of cell types that we want to image and to want to measure and spatially profile requires or demands a high multiplex system to deliver this type of a spatial readout. And so traditionally, this has been done with optical techniques like a microscope for IHC, or even more recently, multiplex immunofluorescence, but it really doesn't solve the problem fully like the way that we want to do. MIF can be complex because it has cyclical staining protocols that are time consuming. They can be destructive to the sample. They introduce variability, and oftentimes they require secondary antibodies or amplification, and it produces a variable and transient signal. And at the same time, anything that's going to be based on light or, specifically in immunofluorescence, you're going to be limited to the number of different probes you can image at the same time. Realistically, it's only about a maximum of 8 with multiplex immunofluorescence as the emission of those probes overlaps with each other in the visible spectrum. You also see high autofluorescence and background in these imaging methodologies, a limited dynamic range, and all analysis is dependent on a digital image of a transient signal. And so really the core limitation here is these techniques can be destructive to the sample during prep and change what the sample is expressing or what you're measuring. You're going to be limited to the number of proteins you can image at the same time, and you're going to have background problems due to autofluorescence. And so that's where multiplexed ion beam imaging, or MIBI, comes in to try and solve those problems. It's a high-definition spatial proteomics platform that delivers actionable insights, where we can analyze 40 biomarkers at true subcellular spatial resolution. So we're getting well past that limitation of 8 that we see in immunofluorescence, and can image more than 40 biomarkers at the same time. It provides detailed morphology and spatial context because we have that subcellular spatial resolution. And it's compatible with FFPE and frozen samples and follows a standard pathology workflow. So it's very similar, sample prep is very similar to what you would experience with other techniques such as IHC. How do we accomplish this? Well, multiplexed ion beam imaging, or MIBI, really is the combination of two pretty unique things together. The first is we have a ToF-SIMS system that we build and develop here at Ionpath, and the time of flight nature of the mass analyzer allows us to image multiple species at the same time. Theoretically, an unlimited number of species at the same time, all in parallel. But we combine that with a targeted approach for staining and imaging, where we label antibodies with heavy metal atoms. These heavy metal atoms are the reporter ions that we measure in the ToF-SIMS system. And they're generally from the lanthanide series of the periodic table. Only a couple, I think, that we use regularly are not, but they're heavy metal. They're from the lanthanide series, so we don't find them endogenously in the tissue, so they make good reporter ions that we can utilize. Basically, we take these heavy metal atoms, we conjugate them to an antibody of interest, or a particular antibody, and we know that that antibody will specifically bind to a protein of interest. And then we can use different reporter ions for different antibodies and build a staining cocktail of 40 antibodies that will target 40 specific proteins and use those heavy metal atoms as our reporters. So the workflow here is we start by staining, and again, we use FFPE or fresh tissue. It's stained with the antibodies, each which has a unique metal isotope attached to it. Each antibody will bind to a single protein target, and then we wash away the extra and put it into the MIBI scope for imaging. This is a ToF-SIMS system, and this is the North America SIMS Society, so I probably don't have to explain in detail what SIMS is, but we've got an ion beam that scans across the tissue surface. It sputters away the material that is there. We capture those reporter ions that we attach to the antibodies that we stained with. And each unique ion represents its then protein target in the sample. We can quantify what is there. The ions are detected on a pixel by pixel basis. We can measure the integrated counts for each of the isotopes we've known that we use for labeling. And then comes the downstream bioinformatic analysis, which includes quantitative phenotyping, protein expression, spatial, and comparative analysis. I'm going to talk about that in detail a little bit later on as we go through. But so what is this, what advantage does this have over the limitations of multiplexed IF? Well, like I said, we're using FFPE or frozen sections, and we have standard primary antibodies. So it's a simplified workflow. There's only a single staining step with the metal tagged antibodies, as opposed to cyclical staining steps that you would need to achieve that level of multiplex with immunofluorescence. We don't have secondary antibodies, and we have a stable signal that we can image on that tissue sample for up to a year later on. There's no spectral overlap like you see in immunofluorescence because we are using ToF-SIMS here, and that in turn then allows us to image the 40 unique channels in parallel at the same time. We don't have autofluorescence, and so that really makes this a more simple technique for this application, but actually allows us to get the same spatial resolution or similar spatial resolution with a much higher multiplex nature. So what do we do at Ionpath? Well, we make the reagents, we make the MIBI scope, and then we do some data analysis as well. And since this is a SIM society, I figure I should probably tell you about the hardware and the ToF-SIMS system that we build. It's called the MIBI scope, and the requirements for this really are defined based on the need or the application we're trying to achieve here. We want to enable this highly multiplexed, high spatial resolution protein imaging, and we want to do so to a market that typically isn't used to using mass spec or isn't familiar with imaging mass spec. And you can think of what that is, but most of our customers are in academic or research hospitals, centers like that, that are more traditionally following or using optical methods. And so we need to make a system that matches that performance and such that it's a simple swap over for them to come onto our platform. The requirements for the MIBI scope, obviously we need subcellular spatial resolution if we're going to image single cells and resolve them. We use a xenon plasma ion source, and it's focusable up to about 350 nanometers, which is well above what you need for that use case. Because of that, we have variable tuning and different sets of parameters that users can select from that allow for a higher throughput that still maintains the single cell segmentation with a spatial resolution of about 700 nanometers. We need a parallel multiplexed detection, so that's why we use a time-of-flight mass analyzer here. That gives us a simultaneous detection of the 40-plus metal labels that we may be using in a panel that we would stain the tissue with. We have a 10 to the 5th dynamic range to simultaneously measure those low, medium, and high abundant proteins of interest. And then really what was one of the main keys is a high throughput that we need. Our xenon ion source is very bright, and it delivers enough ion beam current at that spatial resolution I mentioned above to image an 800 by 800 micron FOV in as little as 35 minutes. And that's really important because the market we're trying to bring this technique to is going to be doing large scale cohort studies of patient samples. And that means you're going to have hundreds and hundreds of samples that you want to acquire, a few images to sample each tissue on. And if it takes 24 hours to acquire one of those images or one of those samples, it kind of precludes you from using MIBI SIMS as a viable technique for that. So really by using such a high or such a bright xenon source and acquiring FOVs in as little as 35 minutes, we start to make it possible so that you can acquire hundreds of samples in the course of maybe days or even a couple of weeks. And then we also want it to be simple enough for non-mass spec users to be able to jump on our platform. And so we've developed software that automates most of the instrument setup and QC and maintains its performance over time. We've defined imaging presets that even further facilitate the acquisition setup. Our customers don't have to sit there and focus an ion beam in a column every day, so that's kind of nice. And then the image data is automatically extracted based on the panel of conjugated antibodies that you specified for those samples and uploaded to our cloud visualization software. And so that is what the MIBI scope is, our ToF-SIMS system for labeled protein imaging. Like I said, we have a xenon primary ion beam that we can focus up to about 350 nanometers. We do have 3 different imaging modes, depending on which gives you a trade-off of spatial resolution versus throughput. Our FOV sizes can be acquired up to 800 by 800 micron with the use of a DEM. One of the key features is there's an electrostatic analyzer after the secondary ions are generated and before it goes into the ToF, which filters out polyatomic signal. That increases the signal to noise ratio of our metal reporters because we know what we labeled with, and they're single metal atoms, and anything we can do to reduce organic signal or other, fragments of other pieces, other lipids, metabolites, proteins, anything else that's in that tissue, all that does is just help us with our signal to noise and really focus on the metal reporters of interest. And it does have an orthogonal ToF for decoupled analysis. We get a mass resolution of about 1,000 to 2,000 at M/Z98, which is more than sufficient for imaging single isotopes of heavy metals. And so that's sort of an introduction to our MIBI platform. What I want to do is walk you through a case study of some samples that we ran at Ionpath a few years ago to really demonstrate what does this data look like and how do we go from the tissue sample to the spatial proteomic profiling that I was talking about. And so what I have here are a number of different samples from, a number of different skin samples that from 4 different groups, so 3 that are diseased and 1 that is normal. What we can do, at the highest level is once those images are acquired, we can take a look at a number of different markers in there to get a visualization of the tissue morphology and the structure of the sample. And so here we're showing some pretty basic markers that show us that are targeted for more structural information. After that, we can dive down and we can look at sort of the immune profile that is there and look at CD45, CD31, and look at a number of different markers. Anyone that has been doing multiplexed imaging or even SIMS imaging of a number of different species of interest knows that you can't really look at 40 different things in one image. There just aren't enough different colors for that. So this kind of shows you, as we go through and drill down into even further subsets of immune cells that are present within these samples, that there's a lot of qualitative data in there already by just looking at the number of different markers that we have. And then we can drill down even further into some checkpoint markers that tell us whether the current state of those cells and whether they're actively fighting an infection. Going back to sort of the most general overview here, what we can see is that the tissue structure does vary quite a bit between these different samples. And that's something we're going to aim to capture with some more quantitative analysis of the sample itself. And so the first thing we have to do, first step in that pipeline, is we need to segment the tissue image into individual. The whole point of this is we want to do single cell spatial analysis, so we need to know where the cells are within a particular sample. And so we segment the tissue image into its individual cell instances, and then we can classify each one of those cells to tell us what type of cell it is based on its protein expression. So that's what you see here. This is one of the tissue images that has been segmented. So each pixel in that image has been assigned to a cell instance. And then based on what the protein expression, so for instance, in red, it is positive for CD3 and CD8. It has we integrate the counts of CD3 and CD8, find that they're above a certain threshold, call those positive, and because it's positive for both CD3 and CD8, it's a cytotoxic T cell. We can do that similarly for other cell types with it that are possible. Of course, they're doing landscaping work right outside of my window right now, so background noise. So we can then take that and we can apply that to all of the images that are in the data set that we acquired. And we see that the populations of these cells differs wildly across the 4 different groups of samples that we have here. And so that's sort of, a primary indicator of, or can be a primary indicator of what's going on in these samples themselves. We can take a look at this relative cell type density, plot that out in a bar or a box plot to compare that across different cell types within the different groupings of the samples. And then we can go ahead and do a comparative analysis of this, in this case, just focusing on the inflamed skin samples, and do a clustering of the heat map to a heat map of clustering to show the similarities and differences between these conditions. And so here we're looking at 4 of the different checkpoint markers: granzyme B, IDO1, LAG3, and PD1, and can see that those have differential those have differential expression across different populations of cells, even within the groupings of samples. And then finally, we can not only can we look at distributions of cells and expression, but because we have the spatial information, we know where each cell is. We can then start to take a look at where those cells are in relation to other features within the tissue image itself. And when I say features, that could be 2 different things. It could be, where a cell is of a particular population. So let's say a tumor cell is relation to an immune cell, or we could do higher level morphological features that are within the tissue sample itself, such as blood vessels or a tumor immune boundary. And so we can profile doing like a nearest neighbors analysis of each cell, and find out how close they are to, or how far they are apart from different cell types or different morphological features within the tissue sample. Okay, and so now I want to talk about some of the applications of MIBI that we've seen out in the literature over the last few years, as our platforms kind of gain some interest and gain some traction. The 3 that I'm going to talk here have come out of labs at Stanford University. And the first one I'm going to talk about is triple-negative breast cancer. So this is from Liat Karen, who was a postdoc at Stanford a few years ago. She's now moved on to the Weizmann Institute of Science and is furthering the use of MIBI there. But back when she was at Stanford, she published this paper on triple-negative breast cancer, which is an aggressive form of breast cancer, where it lacks the expression of estrogen receptors, progesterone receptors, and HER2, so that's the triple-negative part of it. The cancer progression here depends on the interactions between the cells in the tumor microenvironment itself, and the immune system. And 15 to 30% of patients respond to treatment. And so really the question here is what differentiates those populations? So this is a pretty typical question that you can ask and answer with MIBI, because we can analyze with the highly multiplexed nature, we can analyze the number of different cell types, and we have the spatial information and where they are in relation to each other. So really good use of MIBI here. And so every example I'm going to show kind of follows that same workflow I talked about earlier, where we image, or we stain, we image, then you segment the tissue image into individual cells, and then you classify those cells as positive or negative. For particular cell types, you do cell phenotyping. And so we can see here that at the high level of all of the samples included, that there's we can see the cell phenotypes that are present throughout these samples. Here they're just doing a little bit of a drill down into one of the phenotyped images and looking at the expression that led up to that. And then we can go and look at the composition of the immune cell populations that are within those samples. And if you look at this, for all of the samples within this study, we can see that it varies widely from sample to sample. And so at a high, if we look at sort of just this overall population, oops, I lost you. about that. So if we look at the, in the entire sample study overall, there's a particular distribution of all of the different immune cell types that were profiled with the panel that was used here. But if you break that down and look at it at particular patients, you see that that distribution can vary widely as well, and that's pretty evident from the images. So the question then becomes, is this meaningful and what does this tell us? And so if we look at what, tumor compartmentalization in the triple negative breast cancer, so meaning, how is that tumor sort of structured within the tumor microenvironment? And one of the patients here, patient A, you can see they're doing some clustering and a Z-score here to show the expression of all of the markers on particular cell types. We see that the immune markers are co-localized with each other, and the immune tumor cells are segregated. Or excuse me, the immune markers are co-localized, the tumor markers are co-localized with each other, and the immune tumor is segregated from there. If we look at a similar plot from patient B, we see that there's no significant spatial preference in those. And so if we actually go back and look at the phenotyped image from those two particular samples, we see that there's two very different distributions of tumor and immune cells within those images themselves. Patient A, the one on the left, is what is called a compartmentalized, meaning we can see that the tumor markers in orange, or the tumor cells shown in orange here, kind of form these rafts or these regions that are large clumps of tumor cells, and then the immune cells are kind of kept outside of those tumor regions, and that leads to the name compartmentalized. Whereas patient B over here on the right is an example of a mixed tumor environment, meaning that we don't see those large clumps of tumor cells, and those immune cells then have an opportunity to penetrate those clumps and sort of be all around the tumor cells. And if we actually go and look at that across all of the patients, we can split the tumor compartmentalization into three categories. The first one is cold. So an example up here on the top in which we don't have really any tumor cells present in there, so it can't really be compartmentalized or mixed. But then those that do have those that are tumor infiltrated, we can split into the mixed and the compartmentalized categories. And so we see here that if you plot out and look at the number of immune cells, we can see that they do trend towards compartmentalized for more of them. But what we can also do is then look at the structure of those cells and the function of those cells. And so a similar plot here, where in the top we're looking at PD-1 expression on CD8 positive cells and CD4 positive cells. We can see that those with mixed tumors tend to have more PD-1 positive, CD8 positive T cells than PD-1 positive CD4 T cells. In the middle here, we can see mixed tumors tend to have more PD-L1 positive tumor cells than PD-1 immune cells. And on the bottom, we can see compartmentalized tumors tend to have more IDO positive immune cells than IDO positive tumor cells. But the question still remains, what does this matter? Does this tell us anything about the patient? And it turns out that the immune microenvironment organizes clinical response. And so if we think of those three examples again, cold, mixed, and compartmentalized, they actually in this particular study had survival information about each of those patients. And so when you separate out the compartmentalized and the mixed patients and take a look at prognosis over time, we see that the tumor structure can also be correlated with patient outcome. And you see here that compartmentalized tumors are associated with higher survival. And so this is really an example of not just knowing what the phenotypes of all of the cells are in a particular tissue sample, but the spatial distribution and the spatial structure of those phenotypes within the sample is very important and can lead to prognostic information. So the next application I want to go go over here is on ductal carcinoma in situ, or called DCIS. This is another paper that was published a few years ago out of Stanford. Tyler Rhizome was the primary investigator on that. He's now at Genentech. And so Ductal carcinoma in situ, or DCIS, is a pre-invasive form of breast cancer. Approximately, there are 60,000 new diagnoses every year in the US. Incidence increased over tenfold when with screening mammography. But really what you should know here is that it's life-threatening only if it transitions to invasive breast carcinoma. So DCIS is sort of the pre-invasive form of it, and then it's life-threatening when it transitions into invasive breast cancer. And so it's estimated that 50% of women would not progress to IBC if left untreated, but right now the prospective prediction of progression is currently not possible. And so this study aimed to understand, can we figure out any spatial structures or spatial information that would lead us to figure out, or to know whether DCIS would go into the invasive form of breast cancer. And so here we're doing a comprehensive spatial mapping of breast cancer progression using MIBI. This is just sort of an example of what a normal sample should look like, what primary DCIS would look like, and then if it progressed into invasive breast cancer. We see that there's different morphologies and structures of all of the different types of cells within those different types of samples. And so there's two central questions. How does the structure and composition of DCIS change with progression? And what distinguishes DCIS progressors from non-progressors? And so here, it's a similar workflow to what we've talked about. You've got H&E images. They did MIBI imaging on those with a multiplexed antibody staining panel, as shown here, where they were looking at tumor markers, immune markers, and other structural and functional markers within that panel. Do imaging on the MIBI platform, and then get these 38-plex tissue images. And so we have that 38-plex tissue image, and we need to, again, segment that, find out what cell types that are there. That is pretty standard at this point. We can identify the individual cells within that tissue image, do classification to figure out what types of cells those are. But in addition, in this study, there's also a tissue compartment annotation. So if you go back to one of the intro slides on this project, we saw that there were different morphologies or different structural features between the normal and the DCIS samples. And so here they did a segmentation of the tissue regions into the three different types of morphological features: the duct epithelium, the myoepithelium, and the stroma. And so the question then is, how do the ductal and stromal compartments evolve with breast cancer progression? What cells tend to co-localize or avoid one another? And are they typically found within near ducts, vessels, or stroma? So here we're not just looking at what the cell types are and where they are in relation to each other, but where they are in relation to higher level structural features within the tissue sample. And so to digitize this image, not just in terms of the cell types, which has been done previously, but also to understand the morphology of those high level tissue structures. And so, for example, the myoepithelium, you can see that here we've got the raw signal from the MIBI image. And then we can define parameters that sort of tell you about the shape or the size or the structural features of the myoepithelium. So here they're looking at the space from the duct edge. They also binarize that, measure thickness and the perimeter that was covered by the myoepithelium here, and then reduce all of that down into individual parameters that can be used later on. Then there's also pixel level myoepithelium phenotyping and clustering of that and mapping that back to the image. Basically here, we're taking now they were taking instead of just a feature set that tells you it's positive or negative for the 40 or so different cell types that within the cell, they're adding on features that describe the myoepithelium thickness, continuity, and phenotype. And so if we look at, in addition to that, the myoepithelium, there's the collagen fiber size and alignment that could also be quantified, and in terms of the size and the shape, added as 18 additional features describing the collagen fibers. And then all of that can be put into, combined with the cell assignments, phenotypes, and the functional subsetting spatial relationships. And in the end, there's 1,093 spatial and phenotypic features that describe each sample. And so if we go back to the question of what distinguishes DCIS progressors from non-progressors, we can split those into two different sample groups so that we could train a random forest model here, or random forest classifier. So there was a training group of 80% that was made up of progressors and non-progressors. Then there was a test group, and then evaluate that classifier performance and see that we've got an AUC of 0.78. And then go and look at the features that cause the differentiation between the two groups. And it was found that the top 20 features involve 14 distinct cell populations, and it's heavily predominated by stromal myeloepithelial features. And so spatial organization is crucial determinant of risk, meaning here that it was found that not just the phenotypes of the cells present led to the differentiation between the progressors and non-progressor samples, but it's also the structural features that were added in as well. And so if we look here at the phenotype and structure of the myoepithelium, you can see in this normal example here that there's a thick continuous myoepithelium in green that surrounds the other cells. And so here, if we look at two other cases of DCIS where one's a progressor and one's a non-progressor, the question is which one is which. And interestingly, the progressor is the one that still had a pretty intact myoepithelium, shown here green again, where the non-progressor sort of had one that had been broken down and wasn't fully intact anymore. And that was unexpected. And so why would the myoepithelium breakdown in non-progressors be protective? Or, yeah, progressive. And the working hypothesis here is that the thin broken myoepithelium kind of allows the immune cells to come in and figure out that something's going on and protect it from reoccurrence, whereas a dense, intact myoepithelium prevents the tissue from realizing that there's a pre-invasive form of breast cancer here and coming in and fighting this. And the final application I want to talk about is one that's recently come out in a preprint out of Stanford. In this case, we're looking at microglial cell states in brain tissue. Brain tissue is one that's particularly hard to image with fluorescence-based techniques because of the autofluorescence issue that I talked about earlier. And so really MIBI is suited well to try and understand the morphology and distribution of cells within brain tissue better. Microglia are the macrophages of the brain. Unlike immune cell types, microglial cells functions, they can't be easily bucketed based on protein expression, like we saw back in the triple negative breast cancer and the DCIS examples, where we use proteins to say positive or negative for a particular cell type, and that tells you something about whether that what that cell's function is. Here, protein expression of microglial cells doesn't explicitly dictate a cell's function. The function of microglial cells ranges from less active to more active, and that function level varies in different regions of the tissue. So here, not only were they imaging here they were imaging different areas of the brain to assess how this activity state changes in different regions of the brain itself. And see here, they used a 38 marker panel. Again, looking at structural markers there, and then other markers that are indicative of the cell types, especially the microglia and the myeloid cells, which is a big chunk of what the panel is. And then there was imaging on the MIBIScope platform. And so here is an example of the human hippocampus that was imaged. In this case, they actually did a tiled acquisition, so a large scale number of FOVs that were stitched together to reconstruct the tiled image. And we can see that, just by looking at the protein markers here, we can see that expression varies across different regions of this brain tissue. In addition, so then they were able to break that down and segment that tissue into sort of higher level regions of the different areas. And as expected, we can see that the expression of all the markers in the panels varies based on which region or sub-region it's in. But going back to really what was trying to be understood here is, again, that microglial cells cannot just be their function can't be determined based on the proteins that are there in easy buckets. Rather, it needed to be described in a more continuous way. So over here on the left, we see a dimensionality reduction UMAP of all the cells within a particular sample. And we see that there aren't distinct populations of microglial cells based on protein expression. Protein expression varies more across a continuum than distinct buckets for cell types, like I said. But if we define cell types across a spectrum of activity rather than attempt to bucket them into distinct cell types, we see that the activity does progress from left to right. And so that's what this microglial state continuum is trying to demonstrate here, is that it's more of a spectrum of less active to high active cells. And if we plot that back over top of the UMAP, you can see that from left to right, the UMAP is picking up on that as opposed to distinct clusters of cells based on the protein expression. In addition, that continuum of activity varies across the different subregions of the tissue. So those that were those were the segmented earlier. We can see that MSC sort of varies based on where you are on the tissue as well. Here, the authors did pixel clustering to identify subregions of the hippocampus region itself. And you can also see that the microglial state continuum varies differently based on which subregion you are looking at in the hippocampus sample. So here, MIBI captured the microenvironment, spatial proteomic detail throughout the brain. And like I said, human microglia exist on a phenotypic continuum, not as discrete populations. So hopefully those three examples kind of show you what MIBI can be used for, but more specifically, what type of data or what type of spatial information that users or researchers are trying to get out of particular samples and how MIBI can deliver that. What I want to spend the last part of this webinar on is MIBI Plus, which is a new offering from Ionpath that was just released last week. So we're all pretty excited about that. And in this case, MIBI Plus is a spatial proteomics all-in-one service that's powered by our MIBI scope. So what does this mean is that we will take you from start to finish of analyzing your tissue samples, where basically we will stain them, we will image them, we will segment, we will do the data analysis, and we'll provide you back with much of the same information that these researchers were publishing on in the applications that I highlighted. So our MIBI Plus offers a standard human IO panel that has 27 markers and the option to add on 3 additional advanced checkpoint markers. And what does that give you is that gives you 47 single cell phenotypes. So going back to what I was talking at the beginning, this is what differentiates us from other light-based techniques, where we, in a single image, can tell you 47 different cell types that are present in that sample. And in addition, we can identify 6 tissue regions or higher level morphological features, such as the tumor immune boundary, the stroma, lymphatics, blood vessels. So you get a lot of spatial information out of these images, and it follows the same workflow that I talked about earlier. Which is pretty standard. In the case of MIBI Plus, we actually have a online application where you can go and you can place your order for sample kits. We send them to you. You section your tissue onto the sample kits and return them. We then handle the staining. We will then image them on our MIBI scope systems at Ionpath, do the single cell segmentation, the quantification, the phenotyping, and then all of the spatial analysis that comes with that. And so the process for taking that data and getting the spatial bioinformatics out of it is pretty remarkable of what we've developed here. We start with raw data and do some background correction on it, but then we have trained ML models to do the single cell segmentation and the cell classification, as well as some final denoising that needs to be performed on the images. And in the result then are these single cell data sets of marker intensities, protein expression on all of the thousands of cells that are present in a single image. And that ML-based classification workflow is actually is something that we've been developing and are now starting to use, where we train models based on not just where the boundaries of cells are, and then how many how many counts of particular markers there are within that cell to classify it as positive or negative for a given phenotype. But it also takes into account the morphology of, or the distribution of those marker counts throughout the cell. And so here it's showing 6 examples of cells where the top 3 are positive and the bottom 3 are negative. We can see that each one of them is showing here in yellow what would be the membrane marker that distinguishes this as a T cell, where it's present across basically the entire membrane, as opposed to the examples where it's negative, where it's only showing up in a little part, and that could be for a number of reasons. We're looking at a cross section, so it could have been a cell that was above that kind of got cut off when you sectioned your tissue, or it's being contributed from a neighboring cell. But basically, we annotate by hand for a few of these cells to call them positive or negative, train the custom models, and then what you get out of it is a probability that that cell is positive for that marker, and therefore you can do the phenotyping and call it, in this case, a T cell. So here's an example of the classification results you could get. Once the models are trained, results on any new data set is ready to go in minutes, so it's pretty quick. Here you're seeing sort of the raw MIBI images on the top. In A, we have thymus. In B, we have, C, we have head and neck squamous cell carcinoma, and same thing in E and F. And then in the bottom image, you're looking at those images, but they were segmented into single cells, classified based on the probability returned from the ML model. And so you can see all of the different cell types that are there. And then you can also then relate this back to an H&E image. And so this is really nice to show you some of the more traditional methods where you in H&E, where you're only looking at 1 or 2 cell types in a given image. But here over the analyzed image from MIBI, here is showing 1, 2, 3, 4, 5, 6, 7, 8 different cell types in just itself. And we know that that is a subset of the cell types that were imaged in this particular sample. So the innovative bioinformatics helped to distill MIBI's wealth of protein data into cellular identities and functions, which enables a deeper understanding than other traditional methods. Once we have that data, we can follow some of the same workflows I've shown earlier. You start by looking at sort of a clustered heat map of all of the different cell types within the cell's populations. Here we're actually showing an example where there were 2 different types of samples that were in 2 different cohort groups. And so you see here, those that are colored purple are from one of the groups, and those that were colored green are from a different group. You can see that there's a clear difference in the distribution of the cell types within those 2 population groups. We can drill down further and look at the lineage markers, so the immune markers and their subtypes, and see how those populations are different across the different patient types, highlighting here potential differences or similarities in the immune landscape between either individual samples or between screening cohorts. We can do dimensionality reduction and give you a comprehensive visualization of how closely related different immune subpopulations are based on their expression. And so that's what we're looking at over on the left. Here you've got a UMAP where the different point colors here are showing cells of different phenotypes. On the right, we're looking at the same UMAP, but the coloring here shows you the PD-L1 expression within those cells, and you can see which cells and in which of these in the projection do they have high or low expression of PD-L1, which is the checkpoint marker. So the proximity of immune phenotypes reflects functional similarities and potential interactions in the tumor microenvironment, and then mapping those checkpoint markers and their expression across the immune landscape can help decode drug mechanism of action. We can also see that if you splitting out in this particular case, those 2 subgroups I was talking about is a pre-treatment and a post-treatment. So samples were collected initially, they were treated for something, and then they were samples were collected after, and then you can compare the distribution of or not the distribution, but the relative number of cells, or the relative distribution of those cell types pre and post treatment, and see what changes in those samples after treatment and the phenotypic shifts. Post-treatment provides direct feedback on drug impact and other potential unintended effects. And then finally, we can look at the spatial distribution of those immune cells and how those differ between the screening and the treatment samples. And so here we can see that, for each of these 4 cell types, cytotoxic T cells, activated cytotoxic T cells, memory cytotoxic T cells, and naive cytotoxic T cells, the relative spatial distribution of those in proximity to other cell types is different between screening and treatment. One other thing we do offer is a model-based tissue quality score. So we use many of the same models that we use for segmentation and phenotyping to also help us assess the tissue quality and the sample quality itself. This allows us to feel confident in reporting those results when we do go through all of the work to segment and phenotype and then produce all of these quantitative data sets. We also offer interactive classification. So the main output from the phenotyping model is basically a probability for each cell that it is positive for a given marker or for a given phenotype. So we'll allow users to adjust the threshold that is used to classify those, and then regenerate plots and generate a report that's with all of those, the quantitative data analysis on the fly, basically. So we're putting it in the hands of users to take control over their data in a much more simplified way than kind of just handing them a CSV file with tens of thousands of cells in it. And we'll actually even have an interactive tool to be able to generate these plots and play around with them as well. So that's the webinar on MIBI and what Ionpath does. I do want to, acknowledge a number of the people that have worked on this platform and the data analysis in here. Those examples I showed were, I did not do that work. They came out of groups at Stanford, so I need to acknowledge them. And I would be happy to take any questions you all may have on Ionpath, on MIBI. If you want more information on MIBI and what we do, feel free to find me on LinkedIn or send me an email.